Hi. So the long axis of the hippocampus, which in humans runs from the posterior to the anterior ends of the hippocampus, has long been the focus of neurophysiological study. Notably, anatomical tracer studies have suggested that the anterior and posterior hippocampus are connected, via the entorhinal cortex, to different portions of cerebral cortex. And this differential hippocampal cortical connectivity along the long axis has since been observed in functional connectivity analyses as well. And in addition to these differences in anatomical or functional connectivity, the functional diversity of the hippocampus has also been extensively characterized, including the differential involvement of the anterior and posterior hippocampus in processes like memory, spatial reasoning, and novelty detection. And we wanted to re-explore these functional dissociations in the context of broader functional connectivity networks with the resolution afforded by precision within individual neuroimaging. To do that, we studied both hippocampal functional connectivity to cerebral networks and functional specialization along the long axis in two independent cohorts. The initial study consisted of 9 participants that were each scanned extensively over 4 days, while the independent replication set consisted of 11 additional participants, this time scanned over 8 days. And all the analyses I'll show, from hippocampal region identification to functional response characterization, were first completed within each individual in order to respect their idiosyncratic anatomy, and only then pooled to create group averages. So we started in each participant by characterizing their individual networks on the cortical surface. Shown here in red is DNA, a fractionation of the canonical default network, and in blue is a network consisting of both the previously described salience and parietal memory networks, SALPMN for short. These networks were chosen as networks of interest based off of recent work by Zheng and colleagues describing the differential hippocampal functional connectivity to topographically similar networks. So we next calculated the functional connectivity of these networks to the hippocampus, assigning every voxel in the hippocampus to one of these two networks using a winner-take-all approach as is shown on the right. SALPMN1 voxels are blue, and DNA1 voxels are red. Next, we removed ambiguous voxels with either generally low correlations or very similar correlation strengths to both of the networks. And finally, identified the point along the long axis where one network's assigned voxels start to become more prevalent and the other network's assigned voxels less prevalent. We designated that point as the anterior-posterior border, and then assigned each network to whichever side of that point contained the majority of that network's assigned voxels, keeping only the voxels the assigned network had won. Importantly, this process did not assume or enforce that a specific network be assigned to the anterior or to the posterior hippocampus. Using this approach, we replicated the results reported by Zheng and colleagues. In all of our participants, in both the initial and replication datasets, DNA was preferentially correlated with the anterior hippocampus, while SALPMN was preferentially correlated with the posterior hippocampus. Due to the consistency of this finding, moving forward, I'll be referring to the SALPMN correlated region as the posterior hippocampus and the DNA correlated region as the anterior hippocampus. But it's important to remember that these regions were primarily defined based on functional connectivity rather than hippocampal anatomy. And when we explored the functional connectivity of these regions to a core set of cerebral networks in order to assess their selectivity, we found that correlations from the posterior hippocampus, that plot on the left, were highest for SALPMN, the blue bar, while anterior hippocampal correlations, the right plot, were highest for DNA, the darker red bar. And next, when we performed this same analysis in our replication data, we replicated this main finding. You might notice that the overall pattern of correlations from the posterior hippocampus is slightly different, but it's still preferentially correlated with SALPMN, while the anterior hippocampus continues to be preferentially correlated with DNA. So with these data verifying our two hippocampal regions' differential functional connectivity, we next explored their function. Given that many observed functional dissociations along the long axis involve spatial reasoning, such as differences in spatial memory, navigation, or spatial representations, we decided to take a task we'd originally designed to probe episodic projection and deconstruct it from the perspective of space. So in this task, individuals were instructed to answer a number of questions in the scanner, including those asking about their past, like asking them to contemplate the details of their discussion during the last phone call they made, or asking about their future, like asking them to speculate about the next time that they'll go to the beach with a specific friend. And we also included some control questions about their present and general knowledge semantic information. So we then asked a large number of online participants to answer these exact same questions, and then immediately afterwards report how much they thought about spatial information, like how much they thought about the locations of other people, the locations of objects or places, or about visual imagery more generally. And we found that the extent to which participants considered these spatial features varied significantly even between questions designed to engage the same cognitive process. For example, participants reported not really needing to consider spatial information when answering this question on the left, but consistently reported that they heavily considered all three types of spatial information to answer the question on the right. And conceptually, this internal consideration of spatial information falls within the overarching concept of scene construction. This was first proposed by Demis Hassabis and Eleanor Aguirre in 2007 as a key hippocampally dependent cognitive process supporting both remembering the past and imagining the future, but with ultimately broader applicability. And now, we could use these self-reports as a proxy measure for how much a given question required individuals to engage with scene construction, and explore in our MRI data how activity in the hippocampal regions relates to scene construction. And in both our data sets, we found that signal in the anterior hippocampus was positively correlated with scene construction. Each dot here represents a single question with independent values for each study derived from the pooled MRI activation and ratings from at least 25 online respondents. And excitingly, this association with scene construction was specific for the anterior hippocampus. The posterior hippocampus did not display a sensitivity to scene construction. So because SALPMN includes the cerebral regions of the canonical salience network described by Bill Seeley, to study the posterior hippocampus, we turned our attention from space to salience. Specifically, when we explored activity at the borders between task blocks in our initial study, we found transient increases in bold signal within the posterior hippocampus at these transitions, supporting a role for the posterior hippocampus in the processing of salience. To test this hypothesis directly, we included a visual oddball task in our replication study. For this task, participants were instructed to press whenever they saw the target letter, a rarely presented red K, and ignore all of the other letters. And across all of our participants, whenever an individual saw this salient target letter, there was a transient increase in activity within the posterior hippocampus. You can see on the left that this response is only present in the posterior hippocampus, while this plot on the right, showing the average percent signal change within the posterior hippocampus, highlights how transient this response to salient targets is. So, in conclusion, we started with a replication of results from Zheng and colleagues that the anterior and posterior hippocampus have differential coupling to cerebral networks as identified by functional connectivity. The anterior hippocampus is primarily correlated with cerebral DNA, while the posterior hippocampus is correlated with SALPMN. We also found that the anterior hippocampus is sensitive to the process of scene construction, even during a memory task. And the posterior hippocampus is unexpectedly sensitive to transient salient changes. And this functional double dissociation in the response properties of the anterior and posterior hippocampus is reproducible across two independent datasets. With that, thank you so much for watching.